Welcome to Cliniversity. Welcome to the Phases of Clinical Trials module. As we have already discussed what are clinical trials, and we discussed that there are different phases in a clinical trial, let's see the importance of each phase in a clinical trial. So we have 4 phases in a clinical trial: phase 0, 1, 2, 3, and 4. So final stage, we call it as a post-marketing surveillance. So as we have seen different stages in the drug discovery process, discovery and preclinical are the first stages, and under the clinical trial, we have different phases. If you look into the picture, it is highlighting that the population is less in the first phase, and we have gradually increased the number of subjects in the phase 2. And then we have a big patient pool in the phase 3. So why we are gradually increasing the subjects number is, we don't know how toxic or how good or how bad is our drug. That's why we are giving the drug in different dosages in the phase 1 for the few people. And once the safety and efficacy aspect is studied, then we are improvising that drug and we are providing that to the increased number of subjects. And similar thing is applicable from phase 2 to phase 3. We are increasing the population. So we are starting with a lesser population and gradually we are increasing the population. That is our phase 1, 2, and 3 in general. So NDA approval is a new drug approval form. In case if it is a new drug, before releasing the drug into the market, we would require NDA approval. That is new drug application approval. So let's see about phase 0. So the main aim of phase 0 is to quickly weed out metabolically and biologically ineffective drug molecules and introduce very small drug dose into limited number of human volunteers. So in the phase 0, the drug is given to very minimal people. So sample size is nothing but the group of subjects involved in a clinical trial. So what is our target? For how many people we are going to give the drug molecule? So in the phase 0, the sample size is very less. It is between 10 to 15 people only. And we are doing micro dosing in this phase. We are trying to give the minimal dosage to the human participants. So after your preclinical trials, phase 0 is giving the micro dosaging to the human volunteers. And we are checking the pharmacokinetic and pharmacodynamic properties of this drug molecule on these human volunteers. That's our phase 0. And because this is the first phase where we're introducing the molecules into the human volunteers, we can delete or we can remove, we can weed out the ineffective drug molecules in the human subjects. So we can do that in the phase 0. And we have phase 1 trials. So safety is the primary aim. So we are going to check the safety aspects of the drug molecule. And the secondary aim are tolerability, how and what amount of dosage is tolerable in the human volunteer. And what are the pharmacokinetics and pharmacodynamics? We discussed it earlier, how a body is reacting to the drug and how the drug is reacting to the body. So the list of ADME properties and also the drug dissolving properties, pharmacokinetic and pharmacodynamics of the drug. And we are also introducing the drug in the low dosages. So in phase 1, we have two things. One is phase 1A and phase 1B. In phase 1A, we have SAD. We call it as a single ascending dosage. If you see the graph, if you see the table that is highlighted in the right side. So in this example, we have 4 groups and the study is for 4 weeks. So the SAD study, so group 1, the entire group 1 subjects are getting 0.2 mg of the drug in the week 1. And once the results, the tolerability, pharmacokinetics and pharmacodynamics were studied for this 0.2 mg drug, and then we are jumping into 0.4. We are slowly increasing the dose. So we are slowly increasing the dose, and in the week 2, we are giving 0.4 to group 2 people, and gradually we are increasing the dose on week 3, and we are increasing the dose on week 4. And we are studying the properties of this particular drug molecule on human volunteer on each week. So that's how our single ascending dose works. So giving same dose to the different group of people and gradually increasing the dosage. That's our single ascending dose. So coming to multiple ascending dose, MAD, the phase 1 MAD study. So if you see in the right side table, so in the week 1, there are 4 different groups and each group is getting one set of dosage, 0.2 mg for the group 1, 0.4 for group 2, 0.6. So on the same week, week 1, we are giving a standard dosage to this 4 group of people. And we are increasing gradually increasing the drug dosage for all the group people in the week 2. But when you see in single ascending dose, this is performed for each group of people and each set of dosage. But in the multiple ascending dose, we are doing it multiple times and we are ascending. We are gradually increasing it for the each group. So that's single ascending dose and multiple ascending dose. And the main reason to perform this is to study the lower and higher value dosages that we can give to the human subjects. And it is very important to study in this way because we'll get to know what is the maximum and what is the minimal dosage that can be given to the human subjects. So phase 1 involves healthy volunteers and always exceptional cases with oncology trials and chronic trials that are HIV. So in the phase 1 and phase 0 trials, we are choosing healthy volunteers. We are not choosing the diseased subjects. The main reason to choose healthy volunteers, in the phase 0 and phase 1, we are trying to find out the dosage, the safer dosage for the subjects. So we are not checking, how much dosage is curing the disease. We are checking how much dosage will be available in the human body. So that's why it's always preferred to give in the healthy volunteers than your diseased subjects. But your phase 2, phase 3 involve diseased subjects because phase 0 and phase 1, the main aim of these phases is to fix a dosage, fix the dosage. So the sample size for phase 1 will vary from 20 to 100. An alternative name of this phase is called dose escalation studies because we are trying to finalize the dosage. So normally these are unblinded and open label because no disease subjects are involved and this is not a treatment. We are trying to see the dosage patterns. So this is not a treatment trial. So phase 0 and phase 1 are to fix the dosage for our phase 2 and phase 3 studies. So we have phase 2 trials. So the main aim of the phase 2 trials is the efficacy. How efficient is the drug? And safety is the secondary objective. So safety and efficacy are the objectives of our phase 2 clinical trials. And we will find out the optimum dosage because here we are using a huge number of population. So we have a sample size of 20 to 300. So the number of subjects will be involved in phase 2 trials are between 20 to 300. And the alternative name of this phase is therapeutic exploratory studies. And we have diseased subjects in this particular phase 2 clinical trials. And the main aim is to oversee the safety and efficacy. How safe? What are the side effects? What are the adverse events? What are the adverse drug reactions? And how efficient is the drug? How much amount of time is required to cure this particular disease and how much amount of dosage will be required to cure this. Everything will be studied in your phase 2. And because it is given in a higher population, we can also get a clarification about the fixed dose, the optimum dose that can be used. So these phase 2 trials are randomized controlled trial methods, and this will be followed by this phase. So from phase 2, we are going to have randomization. And as we studied, we are going to have blinded trials as well. Then we have phase 3 clinical trials. So the main aim of phase 3 clinical trials is to establish efficacy of the drug against existing therapy. So for example, if there is already drug A, which is used for diabetes, and if you are conducting a clinical trial for drug B, so we are going to have a comparator. So we are going to prove that our drug is similar to the same drug that is used in the market. So it can be benefited if you are releasing the drug into the market. Or the drug that we are using, the drug B, is overweighing the benefits than the drug A. So phase 3 trials are always to compare the efficiency and the safety aspects of the current drug with the existing molecule. So how efficient is your current clinical trial drug against the existing therapy? So again, here we have a large number of population in phase 3, because this is the final, a phase where we are going to analyze and then look into the safety and efficacy aspect of the drug. And here we do have two different phases. One is phase 3A and we have phase 3B. So phase 3A is to get sufficient and significant data. So data is more important in clinical trial. How safe is the drug? How efficient is the drug? How is the efficacy? What is the growth patterns? What is the graph of the dosage and also the reduction of the disease? Everything will be studied in this phase 3 clinical trials. And once we are ending up with the phase 3 clinical trials, we also perform phase 3B that allows the patients to continue the treatment. And when we are getting a good positive results of this drug molecule, we are going to expand the label. So during a clinical trial, we are going to use the label called clinical trial label. So on the particular investigational product, it will be highlighted saying that this will be used only for clinical trial purpose. That will be the main highlight of the clinical trial medicine labels. So coming to market level label, again, IP labeling has to be designed and IP labeling has to be performed. And in case if there is any additional expected or unexpected adverse events. So during a clinical trial, we do have something called an unexpected adverse drug reactions and adverse events. So we might not have a picture that this particular adverse event. So for example, in an oncology trial, so or let us take a diabetic trial. So in a diabetic trial, so weight gain was not expected. Okay, when the clinical trial was conducted, we didn't get any side effect with the weight gain. But when we are giving the same drug in a higher population, so we started observing that there is a huge weight gain issue because of the drug. So now the weight gain, which was not seen in the phase 2, is going to be an unexpected adverse event that has happened in the phase 3. So we are going to look out for this additional safety data where we are going to find additional adverse event or adverse drug reactions ka data, because we are using more and more population in this phase 3 clinical trials. So the sample size will be around 300 to 3000, or sometimes it will go to 10,000 as well. So it depends on the therapeutic area and the particular indication and the efficiency of the drug molecule. The sample size of phase 3 will vary. So it can be 3,000 or it can go to 10 to 12,000 as well. If it is a vaccination trial, the minimum amount of data that we would require will be around 12,000 again. So alternative name of this phase is therapeutic confirmation, and phase 3 is conducted in multicentral level. So for example, same clinical trial can happen in India, Russia, Germany. So this can be a multinational and multicentric trial. So you will have more number of sites compared to your phase 3. So obviously, when your sample size, the number of subjects needed for a clinical trial is increasing, you need more hospitals, you need more centers. And if you're performing the same clinical trial in different countries, we'll get to see about the different adverse reactions that are happening with the drug molecule, and which is very beneficial in the clinical trial. So this is the last in-house phase of a clinical research. So after phase 3 is completed, we have a phase 4 molecule, phase 4 stage, which we call it as a post-marketing surveillance or also a comparison trials. So this phase 4 clinical trials mainly involves safety surveillance, and this will be conducted nearly more than 50,000 patients. So they going to determine the behavior of the drug in the real life situations. And in phase 4, we are going to evaluate action of drug in a situation of unexpected ADRs. So if you see in phase 3, we had 300 to 3000, or sometimes it will go to 10,000 as well. If you do phase 2, we have 20 to 300. So coming to your phase 4, we have thousands of subjects. So we are going to see more number of adverse drug reactions and adverse events. So there will be an unexpected ADRs that was not seen during your phase 3 and your phase 2 clinical trials, because phase 4 is given in a higher population. Because by phase 3, we would have set a benchmark for the safety and efficacy. We would have studied how safe the drug is and how efficient the drug is. But in the phase 4, we are going to look for the unexpected safety aspects that were not seen in the phase 3 and phase 2 studies. And the alternate name of this phase is post-marketing surveillance or post-marketing studies. So pharmacovigilance activities will be conducted in this phase. It is mainly focusing on the pharmacovigilance. Pharmacovigilance is nothing but safety, the safety aspects of the drug molecule on a higher population and also in the real life situations. So let's see the overview, the summary of all the phases and how they are differing from each one. So if you see in phase 1, let's first rule out the sample size. In phase 0, we have 10 to 15, and phase 1, we have 20 to 100. And in phase 2, you have 20 to 300, and it might go to 500 sometimes. And in phase 3, we have till 3000, and it might go to 10,000 as well. And in phase 4, we have more than 50,000 subjects. Let's see the main aim comparison. So in phase 0, we are introducing very small drug dose into limited number of human volunteers to quickly weed out drug candidates or drug molecules that are metabolically and biologically ineffective. And in phase 1 is to assess the safety, tolerability, pharmacokinetics, and pharmacodynamic properties of the drug. Low doses of the drug to a fewer patients. And we have two kinds of studies. One is SAD, single ascending dose, and we have MAD, multiple ascending dose. So your phase 0 and phase 1 is having healthy volunteers, not the diseased subjects. And in phase 2, the main primary objective is our efficacy, and our secondary objective is safety. And we are going to finalize the optimum dose for the clinical trial. And in the phase 3, the main aim is to establish efficacy of the drug against the existing therapy in large number of patients. And the methods, we have two methods. We have two stages actually. First stage is to get sufficient data, and second stage is to follow up. The patients are continued and we are going to follow up, and we are expanding the label from the clinical trial usage to the real world usage. And in phase 4, post-marketing surveillance studies, it involves safety surveillance and it determines the behavior of drug in real life situations. It evaluates action of drug in a situation of missed dosage or overdosage because we are giving it in a higher population. So what are the other names given? Microdosing is for phase 0, dose escalation studies, and phase 0 and phase 1 are all, most of the time, 100% they are unblinded. And phase 2 and phase 3. Phase 2 are randomized studies and phase 3 are also randomized studies. But phase 3 is mainly to confirm the therapy, to prove that, yes, this drug molecule can be used further and can be released into the market. Let's see the guidelines in clinical research. ICH, International Council on Harmonization. So ICH is the website where you can look out for your good clinical practice. So when you type International Council for Harmonization in Google, it will take you to this website. That is our ICH website, International Council for Harmonization. So this is launched in 1990, and group of regulatory authorities from different countries like our European nations, Japan, United States, India, were being part of this conference. So this ICH has conducted a conference, and to that conference, many multinational companies have been attended. So when they have attended, they have finalized a guidelines, and that guidelines are called as good clinical practice. So good clinical practice will fall under efficacy guidelines. So there are different type of guidelines under ICH, International Council for Harmonization. So the regulatory harmonization is achieved by developing guidelines, and these guidelines are divided into 4 categories. The first one is quality guidelines, and the second, safety guidelines, and efficacy guidelines, and multidisciplinary guidelines. So quality guidelines are defining relevant thresholds for impurities, testing, and a more flexible approach to pharmaceutical quality based on GMP, good manufacturing practice. So most of our drug discovery people should follow the quality guidelines. So safety guidelines. So ICH has produced a comprehensive set of safety guidelines to uncover potential risk like carcinogenicity, genotoxicity, and reproductive toxicity. Efficacy guidelines. The work carried out by ICH under the efficacy heading is concerned with the design, conduct, safety, and reporting of a clinical trial. And under efficacy guidelines, we have good clinical practice. Multidisciplinary guidelines, those are cross-cutting topics which do not fit uniquely into one of the quality, safety, and efficacy. So whichever is not fitting into these 3 were put into multidisciplinary guidelines. So let me quickly give you a look on this different guidelines. So let me take you through the website. So this is the ICH website. And if you open ich.org website, you can see there are QSEM. There are 4 different guidelines. So if I opt to open for the quality guidelines. So under quality guidelines, we have Q1. It is starting with Q1 and you have till 14. So there are 14 guidelines under quality, which mainly looking into the GMP. And under safety guidelines, we have almost 12 guidelines, which our main aspect is to look out for the carcinogenicity, genotoxicity, and reprotoxicity of the drug molecule. Coming to efficacy guidelines, there where we can see the exact guidelines. Though there are quality and safety guidelines, these are not completely applicable for clinical trials. So even efficacy guidelines have somewhere around 22 guidelines. So in this, the E6, the main E6 guideline stands as good clinical practice. So this is the good clinical practice guidelines. And once you click on this E6R2 addendum, it will take you to the International Council for Harmonization, Good Clinical Practice. This is the document for good clinical practice, where you have everything about your clinical trials, the principles, responsibilities of IRB, IEC, investigator, and also different aspects in the clinical trial. And as of now, because the first revision was established in 1996, that's why we call it as an R1 revision. E6 stand for 6th guidelines in the efficacy guidelines, and R1 is the revision 1. And this R1 is revised in 2016. It was revised to R2. And the currently used guidelines are ICH, GCP, E6, R2 guidelines. So International Council for Harmonization, Good Clinical Practice, E6, Revision 2 guidelines are the current good clinical practice guidelines that we are using. And E6R3 is under revision. So we have draft guidelines. So they are planning to release this revision 3 guidelines down the line. It might take up to 2 to 3 years to finalize this revision 3 guidelines. So you can download the good clinical practice document from here, and you can see the updates related to the efficacy guidelines anytime in this website. And we do have multidisciplinary guidelines. So it has MedDRA, electronic standards, and we have 15 guidelines under multidisciplinary guidelines. And this is the ICH website. And it's available in it's an open public platform. You can anytime go ahead and look out into this International Council for Harmonization. Let's go back to the presentation. So as I've shown, we have 22 guidelines almost under efficacy guidelines. And E6, good clinical practice. So good clinical practice is an international, ethical, and scientific quality standard for the designing, conducting, performance, monitoring, recording, auditing, analysis, and reporting of the clinical trials. So clinical trials are not just conducted, they should have a good performance, and there should be different parties who has to monitor how the clinical trial has been made and has been performed at the studies, and how the recording, each aspect of the clinical and how it is recorded. And there will be different parties who will be auditing the clinical trial to see and analyze how the data has been collected and how everyone in the clinical trial are working. Are they working as per the good clinical practice? Are they working as per the sponsored standard operating procedures? Everything has to be looked out. And GCP ensures that the data and reported results are credible and accurate, and that the rights, integrity, and confidentiality of the trial subjects are protected. So the rights, safety, integrity, confidentiality is most important. So I cannot reveal that someone called someone like an Afreen or any person whoever is participating in a clinical trial, we cannot enclose their confidentiality. It has to be highly confidential. We cannot reveal everything whatever is happening in the clinical trial. The data should be confidential. But our regulatory authorities and ethics committees are very much aware like how and what is happening in the clinical trial. So this GCP protects those in a trial, but also those whose treatment will depend on the data. This essentially ensures patient safety and credibility of the data. When I say credibility of the data should be relevant. Okay, anything cannot be the data. The data should be the real data. So you cannot put a fictional data and you cannot say that it is a clinical trial data. It should be a real data. If subject says that he had experienced a side effect, there should be a root cause why the side effect has happened and what treatment is given, what is the progression, and how this side effects is interrelated with the drug, and whether there is a relationship or not with the drug, everything will be studied. So you can download E6R2 document from the official website. I have already shown you how to navigate. Once you go into the ICH website, if you go under the efficacy guidelines, you can look for the document under E6 guidelines. So what are the different sections in the ICH GCP? We have glossary, we have principles of ICH GCP, we have IRB IEC responsibilities, we have investigator responsibilities, sponsor responsibilities, clinical trial protocol and protocol amendments, investigator brochure, and also essential documents for the conduct of a clinical trial. So if you look into the content in the good clinical practice, so the glossary, principles of ICH GCP, IRB IEC responsibilities, investigator responsibilities, sponsor responsibilities, and in the sponsor you will get CRO as well, and clinical trial protocol and protocol amendments, like what should actually a protocol consist and what are the list of items that has to be highlighted in the protocol. And then we have investigator brochure. Then we have investigator's brochure. So investigator brochure is nothing but it will have the clinical and non-clinical information of the investigational product. So for example, if you're buying any dermatology product or any such medicine, sometimes your medicine will have a leaflet. So that leaflet is going to give you more explanation about the drug, because the drug might be little sensitive compared to any other drugs. So in generally for some drugs, there will be a leaflet which have a elaborative explanation of this particular medicine. So likewise, in a clinical trial, it is very sensitive because we don't know the complete properties of the drug molecule. That's why we need to have an investigator brochure. So this investigator brochure will have details of your investigational product, that is our drug, the clinical and also non-clinical data. So what is the temperature that you need to use, how to use this drug, if you open and if you're keeping it outside for more than 5 minutes, whether it's going to be active or inactive, how to store it, how to use it, how to handle it, everything will be high. It is a leaflet of your investigational product. So it consists the clinical and non-clinical data of the investigational product. And we do have essential documents. What are the essential documents used in a clinical trial? Same thing was highlighted in the presentation. And let's look into the 13 principles of the clinical practice, good clinical practice. So always remember, each employee or each student, whoever trying to learn about clinical trial, they should be aware about the 13 principles. So this good clinical practice is your Bhagavad Gita, Quran and Bible. So whoever is learning clinical trials, they should be aware and they should understand the 13 principles of clinical trial. So maybe there can be an interview question where specifically they will ask, do you know about 13 principles of clinical trial? So the interviewer is not expecting you to answer all 13 principles, but at least you should try to understand. So if you understand all these 13 principles, it is easy for you to answer in the interview as well. So when they ask about clinical practice, if you are remembering about this principle, or if, for example, if you don't remember, but you should at least remember about QSEM, and then, ICGCP falls under efficacy guidelines. So you should have that knowledge of good clinical practice. So let's look into the 13 principles. So ethical principles. So the first principle in the good clinical practice says that clinical trials should be conducted in accordance with the ethical principles that have their origin in the Declaration of Helsinki. We have studied Declaration of Helsinki in the history. So it's saying that there should be ethics that are taken from your Declaration of Helsinki that are consistent with the good clinical practice and applicable regulatory requirement. So whoever is participating, like a data management team, pharmacovigilance team, clinical operations team, whoever sponsors, CROs, site staff, everyone should have ethics and they should follow good clinical practice and applicable regulatory requirements and guidelines when they are part of clinical trials. And the second principle says that the rights, safety, and well-being of the trial subjects are the most important considerations. And this rights, safety, and well-being, saying that the trial subject rights are important and the safety of the subject is important and the well-being of the subject is important. It is not the clinical trial, but it is all about the subject. It is not about the drug, but the safety of the subject is important first. And after, the development of this drug and giving it to the subject is important. So subject safety is the first priority. And subject safety should prevail over the interest of our clinical trial and also science and society. So it's very easy to remember this, so it's very easy to understand. So the only part is how well you are remembering during your interviews. And next it says, before a trial is initiated, forcible risk and inconveniences should be weighed against anticipated benefits. So always your benefit should be more than your risk. If your risk is more, then you're not conducting a clinical trial. There can be a risk, but it should be minimal risk. So for example, let me take like chemotherapy. So a chemotherapy is performed. There is so much of risk. Hair loss is there, puking, blood loss, vomitings, diarrhea, sick, fever, chills, viral infections because of chemotherapy. But still we are doing chemotherapy because there are these many risks, but you will be benefited with the cancer tumor. That's why we are performing chemotherapy, though there are risks. So this says your risk versus benefit analysis should be performed before starting a clinical trial. So you should always look out for the expected risk and the expected benefits, because we don't know what this drug molecule is going to do, right? So the protocol and the study documents should always have this risk benefit analysis. And a trial should be initiated and continued only if the anticipated benefits justify the risk. As I given an example of chemotherapy, you should always, if there are risks, the sponsor should explain whether this risk is okay for this benefit. So benefit versus risk analysis should be made before a trial is conducted. And then we have, so just now I explained about investigator brochure. So the 13th principle is saying that the clinical and non-clinical information on the investigational product should be adequate. So there should be an investigator brochure before you are starting the clinical trial. And that investigator brochure, it should contain all clinical and non-clinical related information of investigational product. So this IB is going to have all the preclinical data, drug discovery data, everything about this investigational product. The pros, the cons, how to use it, what are the temperature, and what are the other medicines that you cannot use when you're taking this medicine. So it is a huge document, and it should consist of all clinical and non-clinical information of the investigational product. And then we have ethics committee requirements. The clinical trial should be scientifically sound and described in a clear and detailed protocol. So when I say clinical trial is conducted, it should not be from my mouth. It should not be verbal. There should be a descriptive protocol. So what is the visit one? How many days that you are going to give medicine to the subject? And how you are selecting the subject? Are you doing any, pre-analysis before selecting the subjects? And what are the risks? Are you taking any sensitive people like pregnant women, or are you taking any kids into your clinical trial? So everything, each details of the clinical trial will be explained in the protocol. How many visits are there? And how you are going to treat in case if there is any safety aspect, how you are going to treat the patient, subject, everything should be in the protocol. So protocol is a detailed document which should scientifically sound, and the clinical trial description, it's a blueprint of the clinical trial. And the next principle, a trial should be conducted in compliance with the protocol. So a clinical trial should always be conducted in compliance, in the adherence to the protocol. And this protocol should receive an approval. So before using a protocol at study site, it has to be approved by your ethics committee. So institutional review board and independent ethics committee, both are called as ethics committees. So they should provide an approval to the clinical trial protocol. So before a clinical trial is conducted, ethics committee should provide an approval. Qualified personals. So even you are attending this certification program to be qualified for a clinical research and clinical trial related jobs. So the medical care given to and the medical decisions made on behalf of subjects should always be the responsibility of a qualified physician, that is our investigator. So any medical related decision for the subject has to be taken by the investigator. So in case if you're joining as clinical research associate, or if you are into any data management role, you are not the responsible person for the subject. It's always investigator. So the physician is our investigator. So he has to take the medical decision of the subject. If subject is not keeping well, the immediate decision has to be made by the physician. That is our investigator. So they're saying that the medical care given and the medical decisions should be by the investigator always. And there are another people who are working under investigators, and we call them as sub-investigators. They can be either dentist or they can be any other medical graduate people, an MBBS candidate. So either our principal investigator or the sub-investigator or any other medical related field who are working closely with the investigator should take the decision for the subjects. And the other principle, each individual involved in conducting a trial should be qualified by education. Nothing but you people. So whoever wants to be part of a clinical trial in any department for that reason should be qualified by education, and they should be trained. That's why we are training you on clinical research. They should be trained and have experience to perform his or her respective task. So in any department, in case if you're joining in further, even one of the principle in good clinical practice is applicable for you as well. Whoever is in part of good clinical practice, they should have a good education background, training in clinical trials, and also experience to perform their particular task. And then we have respect to the subject. So under this criteria, we have freely given informed consent form. So if you remember about our history discussion, there was freely given informed consent form. That's from our Nuremberg Code. This should be obtained from every subject. So without informed consent form, you cannot give an investigational product to the subject. So informed consent form is mandatory. That is the first document that we are going to collect from the subject. And it is freely given. Subject can take this informed consent and go to any other doctor and come back and participate in the clinical trial. And the other principle, the confidentiality of the subjects, the confidentiality of records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. So always the confidentiality towards the subject has to be maintained. And this confidentiality is utmost important, because we cannot just reveal that someone is having a disease and someone is a part of clinical trial and someone is financially getting benefited because they are part of clinical trial. All these discussions are unethical, and we cannot reveal any identity of the subject to other people. And we have quality principles, where it says all clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. So anything that is happening in a clinical trial, for example, if subject sneezed 20 times in a day, it has to be recorded. If subject had a minor headache, recorded. Subject is out of town, is not taking medicine, record it in the paper. Everything has to be recorded. If it is not recorded, it is not done. A verbal communication will never work in the clinical trial. It should always be documented on a paper. So it should be recorded, handled, and stored. The data has to be stored. You cannot throw the data everywhere. So you have the data on my presentation right now. So I've stored the data in a good way. Likewise, the data of the subjects has to be stored, because this data is going to allow us how good the drug molecule is, because we are going to report, interpret, and verify the data that is found and got by the good clinical practice. So it's very important to record and maintain the data. Investigational products should be, because if you remember about the drug discovery, even for the clinical trial, sponsor is going to give the drugs. Those are our investigational products. So the investigational product should be manufactured, handled, and stored in accordance with the applicable good manufacturing practice. So our sponsor, whenever he's saying that he is providing the investigational product to us, he should maintain the good manufacturing practice before providing the drug to the clinical trial people. So before giving the drug molecule to the subject, it should be manufactured accordance with the good manufacturing practices. So without good manufacturing practices, a drug cannot be manufactured. So GMP is very important. And sponsor has to follow good GMP. And this should be used in accordance with the approved protocol. So they need to use good manufacturing practice. Systems and the other principle. Systems with procedures that assure the quality of every aspect of the trial should be implemented. So systems meaning any electronic systems or any instrumental systems that we are using, and with the procedures, how to use the system. So a standard operating procedures for a tool has to be maintained to assure the quality of every aspect of the trial. So that's very important. So even when you go to this document in the ICH, if you click on the principles of ICH GCP, whatever we discussed about the 13 principles, I'm just going to summarize again. So first one says about the ethical requirements. So first one is talking about the ethical requirements of the clinical trial. And second one is talking about the risk versus benefit analysis. And third one, the rights and safety of the well-being of the subjects. And 4 is talking about IB, the clinical and non-clinical information on the investigational product. And 5th one is talking about the protocol. The protocol should be scientifically sound. And 6 is talking that this protocol has to be approved before using in a clinical trial. And physician should be qualified, and the decision about the subject has to be made by the physician. And number 8 is saying that each individual like you and me involved in a clinical trial should be qualified with education and trained. And informed consent form is freely given. A freely given informed consent form to the clinical trial participation. And all clinical trial related information should be recorded and handled. So it should be maintained in a particular document. So the lines here specifies that what has been revised from R1 to R2. So not only here, if you scroll down, you can see this sort of lines, throughout the document. So this says that this part, because this is an R2, this is a revision 2 document, right? So an addendum means change. So this points 4.2.5 and 4.2.6 were not there in R1. So these have been added into R2. So in this R2 document, you will find, whatever the data with margin lines like this says that this has been added into the revision 2. Okay. So there were 3 principles again added into the revision 2. So we had only 10 principles in R1, but we have 3 principles plus 3 principles in R2. So the point number 11 says about the confidentiality of the subject. 12 is talking about IP and good manufacturing practice followed for the investigational product. And the systems, whatever systems and procedures we are using, it should have the quality of every aspect in the clinical trial. So these are the 13 principles that you can see in the document also, and also in the material that will be provided by Cliniversity. So we discussed about different phases in the clinical trial and also about the good clinical practice, the content in the good clinical practice, and the 13 principles of the good clinical practice. Further, let us see about the main clinical research terminologies that you should be aware about. So let's talk about the major clinical research terminologies that are very important. adverse event. So an adverse event is any unfavorable and unintended sign, symptom, or disease. It may or may not be related to the medicinal product. ADR, adverse drug reaction, a response to a drug which is noxious and unintended and which occurs at dose normally used in human for prophylaxis, diagnosis, or therapy, and which has a causal relationship with the drug. So let me give an example of adverse event and also adverse drug reaction. So if a subject has taken medicine meaning like investigational product. So there are two subjects, subject 1 and 2. So one has taken investigational product and 2 has taken investigational product. They are both part of our clinical trial. Now subject 1 was walking on the road and was hit by a bike. And the subject 1 got injured. Subject 1 is having a knee surgery. Okay. So this is called an adverse event because it does not have any relationship with the drug. So the subject was not experienced any nausea, dizziness, nothing. The subject was randomly walking in the road and a bike hit him. So this was an adverse event. So an example, now subject 2, who has taken investigational product, was walking in the home and after taking medicine, subject felt very dizzy. And when he was taking stairs, he fell down and then he injured his arm. So now subject is going to say, I have experienced a dizziness and I fell down. This dizziness is a reaction by our investigation product and it is having a causal relationship with the drug. That is called adverse drug reaction. So if you see in the glossary of clinical research, in the good clinical practice, the first two definitions are about your adverse drug reaction and your adverse event, and elaborative definitions. If you see here, the main thing that you need to keep in mind, an adverse drug reaction will have a causal relationship between a medicinal product and an event, wherein adverse event, okay, an adverse event will not have, okay, an adverse event which does not necessarily have a causal relationship with the treatment. So adverse drug reaction is having a relationship with the treatment and event does not necessarily have a relationship with the treatment. Compliance. So there always might be a question, what is the difference between AE and ADR in your interviews? So you should say that an adverse event necessarily will not have a relationship, but an adverse drug reaction will have a relationship. The name itself says that. And then we have compliance. Adherence to all trial related requirements like being compliant, adhering to the good clinical practice and adhering to the protocol, adhering to the regulatory requirements is being compliant with the protocol. So compliance is very important in a clinical trial study. Confidentiality, we have discussed multiple times. Prevention of disclosure. So you're not disclosing anything about the study or anything about the subject. That is your confidentiality. Efficacy. We have multiple times seen about efficacy. A product's ability to produce beneficial effects on the duration of course of a disease. So efficacy is measured by evaluating the clinical and statistical results of clinical test. So efficacy is the ability to produce beneficial effects. Okay. Producing a beneficial effects is nothing but your efficacy. How efficient is the drug? And in protocol, we have two criteria. So when we say we are selecting a subject, so for this selection, there are two criteria. One is inclusion criteria and the other one is exclusion criteria. So inclusion criteria is a list of criteria that must be met by all subjects. So all eligibility criteria is your inclusion criteria. All non-eligibility criteria is your exclusion criteria. So for example, I don't want pregnant women in my clinical trial. So my exclusion criteria will be pregnant and lactating women are not part of clinical trial. So if any woman coming and if she's lactating or if she's a pregnant woman, she will not be enrolled in a clinical trial. And anyone who is having more than a sugar reading of 350 if there is glucose reading, they are not part of my clinical trial. So that will fall under my exclusion criteria. And anyone between 18 years to 50 years can be part of my clinical trial. So that is my eligibility criteria. That is my inclusion criteria. Anyone with diabetes and with a glucose reading lesser than 300 can be part of my clinical trial. That is my inclusion criteria. So in a protocol, we'll have two criteria, the eligibility criteria to be part of clinical trial and the non-eligibility criteria. This is called as inclusion and exclusion criteria in a protocol. And usually in an informed consent process. So let me quickly explain you about 3 scenarios in informed consent. So if I'm the investigator, okay, and if I have 3 type of subjects in my study. So subject 1 is literate. He can, whatever I'm explaining to him, he can understand. So in this scenario 1, the subject is literate. So this literate subject can understand and sign for himself. So there will be a conversation between investigator and subject on informed consent process. In case if subject is not literate, okay, if subject is not literate, or if I'm conducting a clinical trial for kids, if the subject is under 18 years old, so then subject cannot take a decision on participating in a clinical trial. So in the scenario 2, if the subject is illiterate or the subject is a kid, they would require an immediate family member. So this immediate family member who is taking decision on behalf of my subject. So like your father taking decision on behalf of you in this informed consent process, or your mother taking decision instead of you. It can be vice versa also. If you have an elderly, patient in elderly subjects, if a subject is 72 years old, then his son can be the immediate decision maker. So these people are called as legally acceptable representative. In the scenario 2, where the subject is illiterate or the subject cannot give consent on their behalf. So an individual or judicial or other body authorized under applicable law to consent on behalf of a subject. So on behalf of a subject, if an individual can give a consent, that person should be related to the subject. They are called as legally acceptable representatives. And scenario 3, wherein if the parents are also illiterate, like just an example, if you and your parent, if you both cannot make a decision, if you both are illiterate, then we have a third person called impartial witness. So who can be an impartial witness? Your family doctor, your family nurse, or your family lawyer, or your immediate family member who is educated. They can be an impartial witness. Or a doctor, the investigator also can arrange an impartial witness. And that the impartial witness should not be part of a clinical trial. They should be independent from the clinical trial. So a person who is independent of the trial and who cannot be influenced by people involved within the trial, who attends the informed consent process if the subject and the subject's legally acceptable representative cannot read. So scenario 1, where I am explaining it again. So we have 3 scenarios in informed consent. Scenario 1, if subject is literate and subject is good with the investigator explanation, so there will be investigator and subject conversation and they will be done with the informed consent process. But if subject cannot give the consent, then we would require, if the subject is illiterate or cannot give, then we need subject and legally acceptable representative. So in scenario 3, we have investigator, LAR, and subject. But in scenario 3, if LAR is also cannot read and write, then we have impartial witness. So subject, LAR, impartial witness, and investigator will be part of informed consent form process. So you should read the definition again and again to understand the concept. So please read about legally LARs and also impartial witness to understand, and listen to the explanation that I have given again. So then we have investigational product. An active ingredient or placebo that is being tested or used as a reference is our investigational product. So we have studied about adverse event and adverse drug reactions, right? So we consider it as a serious adverse event or serious adverse drug reaction if that side effect is falling under these criteria. So if an adverse event is resulting in death, then it is called as a serious adverse event. If the adverse event is life-threatening, like any abnormality or if any dysfunction in an organ, if there is a kidney loss, kidney is not functioning, then that is life-threatening. So then that is a SAE. If the subject has to be hospitalized for more than a day or for at least for an hour or 2 hours, that is also considered as an SAE. Hospitalization and prolonged hospitalization, which is resulting in a disability, fracture in the leg or fracture or a brain tumor or a memory loss, anything like that, which is resulting in a disability is also an SAE. A congenital anomaly, a birth defect, which we have seen in the history also. If in a clinical trial, if someone is participating and that is resulting because of the drug, if it is resulting in this birth defect, that is also called as an SAE. Or any medically important event, like they have some psychological disorder or they have a different condition, they're not the normal person. And nowadays, even cancer is also considered as an SAE. So any adverse event, in case if it is a death, life-threatening, hospitalization, a disability, a birth defect, or medically important event, or a cancer, they are called as serious adverse events. Those are SAEs. Any event, any side effect, in case if it is falling into this criteria, that is called SAE. So you need to by heart all these events. So there will be a question in your interview about explain SAE. So you need to say an adverse event which is resulting in death, life-threatening, hospitalization, disability, birth defect, and medically important event. So you should by heart this. So please remember the definition of SAE. And then we have safety reports. So any reports which are related to your adverse events, your SAEs, those are called as your safety reports. Source data and source documents. Source data is nothing but when we say the subject is having high blood pressure or subject heart reading, any medical records or any original records of subjects are called as source data. So all information in original records and certified copies of original records of clinical findings, observation, or other activities in a clinical trial necessary for the reconstruction and evaluation of the trial. So if I say that subject had a high blood pressure, so where should I go? That is a source data. I will have that data in the document. So storing of this data in a document is called as source document. So all your medical reports, for example, if you underwent any, a body test, a blood test. So that blood report, that laboratory reports, your X-ray reports, so of our subjects. So subject data, which is talking about subject health, is our source data and our source documents. And subject, we know about the subject definition, an individual who's participating in a clinical trial or either a person who's receiving the investigational product is called as a subject. Each subject will be given with a subject number that is called as subject identification code. Like how you have registration number for your college, like for your exams. Likewise, each subject will be given with the number. And that unique identifier number is called subject identification code. So we will be calling the people, the subjects with the numbers. We're not calling with their names. Because when you remember about confidentiality, we said confidentiality is important. So we are not revealing the names, but we will be giving them with the code and they will be called with the SIC. That is our subject identification codes. Then we have vulnerable subjects. A group or individual that cannot give informed consent because of limited autonomy. So for example, children or mentally ill, like your autistic children or mentally psychologically deficient, because there will be clinical trials happening for psycho-related drugs as well, psychotic drugs as well. So those people are called as vulnerable subjects. So because they can't take a decision on their own, right? So we call them as vulnerable subjects. So vulnerable subjects also refers to subjects who may be unduly influenced. So for example, students, subordinates, or patients, we can easily tell them, see, there's a free medicine given, why don't you go and take it? So we need to treat vulnerable subjects will have again different way of, explaining informed consent form, and all vulnerable subjects requires a LAR and impartial witness, legally acceptable representative, because they are children and they are mentally ill. They cannot take decision on their own. So vulnerable subjects are very sensitive, and a clinical trial with vulnerable subjects is also very sensitive. Let's start a discussion on informed consent form, case report form, and protocol. So these three are the basic essential documents that are used in clinical trials. And though we have multiple essential documents in the clinical trial, informed consent form, case report form, and protocol plays a major activity coming to the clinical research. So we'll be looking into the informed consent form process and also the CRF template. What are the contents in the informed consent form, introduction to case report form, and CRF template, protocol and protocol contents. So informed consent process. So a process by which a subject voluntarily confirms his or her willingness to participate in a particular trial. So before a clinical trial is started, informed consent is the first document that will be discussed between investigator and subject. So before we start any clinical trial procedures, the first thing what investigator has to do is to collect an informed consent form by the subject. And this informed consent form is finalized by the CRO and sponsor. And investigator will use this informed consent form to get the signatures from the subject. In case if subject is willing to participate, then the subject is going to final the signatures, and then the subject can be part of a clinical trial. If subject is not willing, after explaining everything about the clinical trial, if subject is not willing to participate, then the subject cannot participate, and it is up to his willingness. He can either carry the informed consent with him or leave the informed consent with us, and he cannot participate in the clinical trial. So it's always the subject decision. So after having been informed of all aspects of the trial that are relevant to the subject's decision to participate, so we are providing all the information related to the clinical trial, and subject has to decide whether to participate or not to participate in a clinical trial. Informed consent is documented by means of a written, signed and dated informed consent form. So an informed consent form will have signatures of the investigator subjects. So informed consent document, it is called as an ICD document. So it will have a patient information sheet or a clinical notes. So when a patient is giving an informed consent form, we do have an audio video recording, because the regulatory body and ethics committee wants to make sure what investigator is sharing with the subject. What are they sharing about related to the clinical trial? Are they explaining the pros, cons, the side effects, everything or not. So for this, there is something called AV consenting. We are going to record what investigator is talking to the subject. So in the patient information sheets or in the clinical notes, patient information sheet is also called as clinical notes. So when an informed consent process is happening, the subject might be familiar with the Telugu language or Kannada language or Tamil language or Malayalam. So investigator will not talk in English all the time. So he's going to explain the concept of this clinical trial, and he's going to explain about the informed consent, whatever present in the informed consent form in the local language. So the language specification and the willingness of the subject has to be narrated as a story in your PIS sheets. So your clinical notes will have a description of what actually was happened in this informed consent process. So it will be like this. So investigator has explained, and it will be in English. We are writing it in English, but they would have talking in the local language. So in the patient information sheet, we will have like investigator has explained all informed consent related details in Kannada language, and subject was comfortable in Kannada language. So a Kannada informed consent form was used. Subject has been told about the side effects. Subject has been informed about the duration of this clinical trial and importance of each visit. Subject was willing to participate in the clinical trial, and subject has completed the signatures. Subject will be moved into the screening procedures. So it's a narration of, so this patient information sheet will consist of the narration, actually what happened in the process. And informed consent will have signatures from principal investigator and also from the subjects. And we are also recording everything in a tab. So sponsor will be providing a tab at site. So study nurse or anyone who's working at site can record the conversation of investigator and subject during an informed consent process. If you see the pictorial representation right side, investigator is with subject. So, and he's using an informed consent form. He gives information about purpose of the study, requirements that has to be met, the treatment of the study, and the usage of placebo as well, and how to participate in the study, risk and inconveniences that may arise in the study. So the patient and the doctor signs the informed consent form and the patient information sheet. One copy is saved at site and the other copy is given to the subject. Now, subject can take this informed consent form and go to any doctor, any lawyer, or wherever he want to go. He can show this document to anyone and then take another opinion. So we are not influencing, we are not forcing the subject to complete the signatures. And we are also giving a free informed consent form where subject can go and take his second opinions and come back and decide to participate in the clinical trial. So informed consent is an important process, and it is the first document that will be collected from the subjects. So what does an informed consent form consist? So we're telling that investigator has to explain about the study, investigator has to tell everything to subject. So what are the main things that an informed consent will consist, and what will be explained by investigator to the subject? So investigator is going to explain that the trial involves research and also the purpose of the trial, why the trial is conducted, and the trial treatment and probability of randomization. We already discussed about randomization, right? So the subject will be informed about randomization. And trial procedures. What are the trial procedures? How many visits are there? Are we going to take some blood for a blood analysis? Are we going to collect any urine samples? Are we going to collect any sputum samples in case if it is a lung disease or anything related to that? And the subject's responsibility. So we should also tell, like investigator should explain that subject should not miss any visits. So he should be compliant with the clinical trial protocol. The subject has to come to the hospital whenever he's been asked, and subject has to inform in case if he's experiencing any side effects. Explaining experimental aspects. So what all will happen in this clinical trial? Which visit is important? A clinical trial protocol might have 20 visits. Maybe like, every 2 months once a subject has to come to the hospital and take the investigation product and go back. And reasonably forcible risk. What are the risks associated in case if the subject is participating? We are telling everything to the subject. We are not leaving it. We might tell him that, see, you might experience so-and-so side effect, you might get it, but still you will be benefited. We're going to explain this. We'll explain the risk as well. We'll explain the benefits as well. And the alternative treatment in case if subject is getting any other infection in the course of clinical trial, what is the alternative treatment or alternative medicine that the investigator will be giving? And we will also support in case if subject is having any comorbidities during this clinical trial. And compensation in the event. So for each visit, subject will be paid. We are paying subject because it's part of our clinical trial. So for each visit, for each medicine intake, sponsor will pay to the subject. And what is the compensation? And in case if something bad, like, in case if subject is experiencing a hospitalization or anything like that, the compensation will be bared by the sponsor organization. So they're going to look out and they're going to provide the financial support to the subject. Anticipated payment. And once this clinical trial is completed, subject will be paid with the good amount. And voluntary participation, meaning like subject can anytime withdraw from the clinical trial. It's up to subject decision whether to participate or whether not to participate in a clinical trial. And subject can decide to withdraw at any time. And he should be telling us the reason, that's it. We are not forcing him or we are not threatening him to participate in the clinical trial. It's up to subject decision in case if he wants to withdraw from a clinical trial in between, he can withdraw. We are not costing anything to the subject. It's up to subject's decision. And access to the medical records but maintain confidentiality. If subject wants to show the progression of him in the clinical trial to any other doctor, we are giving him the certified copies wherein we are taking a Xerox copy and providing it to him. He can take this medical records and show to any other doctor, and then it's up to the subject. In case if he requires, he need to write a letter. So the site staff is going to provide this medical records to the subject. And any new information that is related to the clinical trial, if there are any changes in the dosage or any changes in the upgradation of medication, that will be informed to the subjects. And we are also taking an information, like we are taking and giving. So there will be a doctor who's 24/7 available for all the patients. So in case if they're experiencing any side effect or any abnormalities, they can anytime call this contact person. So we are giving a doctor number to the subject, and that person has to be available all the time, 24/7. Termination of the trial. So we'll also explain subjects that in between, if there is any abnormalities, we are going to terminate the clinical trial, and that information, why we are terminating, what are the side effects, it will be clearly explained to the subjects. And total number of subjects, because a clinical trial will not have only one subject. As we have seen about the phases of the clinical trial, there will be the sample size for each clinical trial is different. So we are telling the subjects, you're not the only one. We have already recruited so-and-so subjects into the clinical trial, and we will be recruiting many more in the clinical trial. That information will be given to the subjects as well. And if they want to talk to any other subjects in the clinical trial, we are very open and free to provide their information to the subjects as well. So these are the main contents and explanations that are included during an informed consent process. So now let us look into the case report form. Case report form. So case report form is a specialized document in clinical research, and it should be study protocol driven. So a case report form is designed with respect to the protocol. So protocol is the main document. So by knowing the protocol, the one person who is working in this particular department, who is designing the documentations, like your medical writing department, your operations department, they are going to design this case report form with the use of protocol. So this case report form will consist of robust content and have material to collect the study specific data. In case if the subject is coming, so first we have completed informed consent process, then the subject is randomized, and then the subject is enrolled, meaning like the subject is into the clinical trial. The subject is participating in the clinical trial. So when they are participating, there will be visit 1, where the first visit for the subject, and visit 2, continuously we will have n number of visits in the clinical trial. As per the protocol, the visits will be designed. So in each visit, when subject is coming, we are going to record subject sugar values, blood pressure values, and the any adverse effects, any adverse events if subject has underwent, and what is the progression in any other medicines if subject is taking, or does he have any other concomitant medication, meaning like any medicine that is taken apart from the investigation product. So for example, if it is a migraine study, the subject would have already had thyroid, blood pressure, and diabetes. So just because the subject is taking investigational product, he cannot stop taking other medicines because he already having an existing condition, right? So we will let him take that medicine as well. So that medicines, the other medicines which the subject is taking apart from investigational product is called as concomitant medications. So your case report form will record the concomitant medications as well. So though paper CRFs are still used largely, paper CRFs. So there are two types of CRFs, electronic CRF, that is called as eCRF, and also paper CRF. So though there are paper CRFs, the data which is recorded in the paper CRF will be made available in the electronic CRFs as well, in the electronic case reports as well. So online discrepancy management and faster database log is planned in case if we are using electronic case report forms. So eCRFs are gaining popularity due to the advantages they offer, such as improved data quality, online discrepancy management, faster database log. Main objectives behind CRF development is to preserve and maintain quality and integrity of the data. So when the CRF is completed at the end of the trial and signed by the investigator, the original CRF is sent to the sponsor. The investigator must keep a copy of the case report form, and thus the data generated at the site, so the sponsor does not get exclusive rights over the data. Sponsor cannot edit data without the investigator's approval. So a case report form, so there is an electronic data capture system, which we call it as an EDC system. So all the EDC systems will have eCRF documents. So wherein we are entering the data, the demographic data, the details of the subject, everything that we are collecting in the so-and-so visits. So case report form will record the subject data. And electronic cases, so if you're writing it on a paper, same thing has to be, so data has to be entered into your eCRF. So eCRFs are present in electronic data capture system. So we call it as an EDC systems. So this is an example template of case report form. So patient informed consent, whether it is taken or not. So we need to write. So it's a blank template. We'll be mentioning, yes, informed consent has been taken. So date of participant signed the informed consent form. You're going to mention the date when the participant has signed the informed consent form. Date of first trial related procedures and name of the person taking informed consent. So the investigator name, who's taking the informed consent from the subject. And we have demographic data where we are going to mention the date of birth, ethnicity, like which gender, which category and which region they're going to belong, and what is the sex of the participant, either male or female. And then you will have height, weight of the participant, body mass index, and blood pressure details, heart related details, and anything related to the protocol. If it is a cancer trial, we are going to look out for the tumor analysis, and all this data will be recorded in the case report forms. And it is important to record. And the same similar data variables, we call this as a questions. And same sort of questions will be available in the EDC system. That is our electronic data capture system, where a person from the study site is going to enter the data that is available in the paper CRF and is going to enter the same data into the electronic data capture systems. And this EDC tool can be viewed by the sponsors, CROs, and across the clinical research team, whoever is participating in the clinical trials. So the subject data can be viewed by event sponsor, though they are not part of the site level. So no one can change any data in this CRF document. It is only the investigator who can review and change the data or correct the data in the CRF forms and even in the EDC. There can be multiple queries raised by the different departments across the clinical research team, like why this is recorded, why there is an abnormality. Such data queries can be raised, but corrections should be made by only investigators. So CRF is originated again. CRF will be designed by using your protocol. Now let's see what is a protocol. So a research protocol is a document that describes the background, rationale, objectives, design, methodology, statistical considerations, and organization of a clinical research project. So this particular protocol document will explain you the background, why a clinical trial is conducted, rationale, objectives, what are the primary and secondary objectives, and what is the design, what sort of design they are using. Are they using a parallel or crossover design? What is the design used in the protocol? And what are the methodologies that they are using? And what are the statistical considerations for recruiting the subjects and providing the investigational product? And organization of a particular clinical research project. A protocol is a complete blueprint of a clinical trial. So it consists everything and how, when, why, what has to be performed in a clinical trial. A study protocol must be approved by the ethics committee before starting a trial. So before starting a clinical trial, the protocol must be approved from an ethics committee. And you will get a regulatory approval as well. And you'll also get an ethics committee approval as well. It is mandatory to get an approval. And from this protocol only, we are designing the case report form. So what are the contents in the protocol? Protocol, according to the ICH Good Clinical Practice, a protocol should include the following pages. A protocol should have a title page, and it should have the background information. It should have the objectives and purpose. It should have the study design. Selection of the subjects, like the inclusion and exclusion criteria, eligibility and non-eligibility criteria to select the subjects. And treatment of subjects, what sort of treatments we are giving to the subjects. Assessment of efficacy. So how the efficacy is assessed, how the safety parameter is assessed, how we are going to analyze and assess this sort of safety and efficacy. And how to record the adverse events. In case if there is an adverse event, how to record. And if subject wants to not continue in a clinical trial, what are the process and procedures for the discontinuation of the study? And the statistics, how many, what is the sample size, how many has to receive some treatment, treatment B, C, and how we are going to calculate this, and how adverse events and the drug is related or not related, how we are going to do this, all the statistics of the study. Quality control and quality assurance. How are we going to maintain the quality throughout the clinical trial? Ethics. And what are the ethics that we are following in this clinical trial? And what are the applicable ethical guidelines that we are following? Data handling and record keeping. How the data that is recorded in the case report form should be handled. And how we need to maintain records for this. How many years the record has to be maintained. Publication policy, in case if you want to publish anything related to the protocol, and are we going to publish at the start or in between, and which parties are involved in this publication policy. Publication means publishing the progression of this particular clinical trial. So to publish this protocol or to publish this data, we should need an approval. and project timetable and flowchart. So when we are going to start, and in between, in case if we need to extend, how we are going to do that, and references, through which references we have designed this protocol, and appendices in case if we are using any particular format of data collection, and those examples as well will be added into the protocol. These are not limited. In case if we require to add any other topic in the protocol, we can do it. But these are as per ICH Good Clinical Practice, the mandatory items that should be available for the protocol. We'll also have the rationale. The purpose is nothing but, objectives are nothing but rationale, why a protocol is being used and why a clinical trial is conducted for this particular drug molecule by using this protocol. And as we have already discussed about Code of Federal Regulations during the history of clinical trials, and I have shown you that 21 CFR is specifically for the Food and Drug Administration. So I have shown in the website that Food and Drug Administration is under Title 1. So let me quickly take you through the new portal as well. So let me open this ECRF website for you, ECFR. So Code of Federal Regulations, Code of Federal Regulations. And as we have already seen during the history discussion, Title 21 stands for Food and Drug Administration. So in this, we have 3 chapters. So in these 3 chapters, we are going to see some important Code of Federal Regulations that you need to remember. So the CFR is a codification of the general and permanent rules that were published by the federal regulations, by the executive departments and agencies of the federal government. It is divided into 50 titles and that represents broad areas subject to federal regulations. And it was revised, the entire titles was revised in 2018, and on day-to-day basis, they'll keep on revising the regulations. So 21 CFR is a section in the Code of Federal Regulations that sets forth the United States Food and Drug Administration guidelines on using electronic records, electronic signatures. So each title of the Code of Federal Regulation addresses a different regulated area. So 21 CFR relates to pharmaceutical and medical devices, and Part 11 being applicable to the electronic records and electronic signatures. So let's see, because he's highlighting, like I'm highlighting here in the Part 11, right? So what they're saying, though, it's all about electronic signatures and electronic records. So if you have seen the previous example, when we are discussing about case report form. So when we're discussing about case report form, we have told that we are using EDC system. So EDC system is nothing but electronic data capture systems. So in this system, we are recording the data electronically, meaning like someone has to have login credentials. So how you have login credentials for your social media, like Facebook, Instagram, even the EDC system should have login credentials, and each person will have unique login credentials. And as we multiple times mentioned, the clinical trials are very confidential. So you cannot share login credentials with anyone else. Only you have to log in with your login credentials. So here it is saying that Part 11. So if you see the numbers here, it's 1 to 11. So 11 is coming between 1 to 99. So I'm clicking on this part. And if you go down, Part 11, so 21 CFR Part 11 specifies about electronic records and electronic signatures. So anyone can go ahead and read about this entire concepts of electronic signatures in case if you are interested. Let me summarize. So it specifically says that when you are using electronic signature is nothing but your password. Electronic record is nothing but having the data electronically. Like all the subject data is available in the ECRFs, right? So in the EDC system. So you are having the subject data in this ECRF and you are using an EDC system, an electronic data. So for that, there are some sort of rules and regulations, and each company, whoever is using an EDC system, should follow 21 CFR Part 11. So it is specifically specified as 21 CFR Part 11, and it says that it should be highly confidential when you are using electronic records and electronic signatures. So likewise, we have different parts like 1 to 99, and totally we have 1500 parts, and the title 21, because when this presentation was made, it was 1400, and now we have 1500. So what are the important Code of Federal Regulations that you have to remember? So 21 Part 11 is about electronic records that you need to remember. Then 21 Part 50, it's about informed consent process. So let me go back to chapter 1. And if I go to part 50, so I open 1 to this, and part 50 says about protection of human subjects. So you need to remember 21 CFR part 50 is about protection of human subjects, and it's about informed consent form. And part 56 is about IRB, Institutional Review Board. So part 56 is about IRB, part 56. And then we have 312 about investigational new drug application. So let me go back to this chapter. And it says that 300 to between 499. And I'm going to 312. So investigational new drug application, IND application. And then we have 314. 314 is for approval for FDA approval to applications for FDA approval to market a new drug. So not everything is related to the clinical trials. So there are some basic CFR parts, Code of Federal Regulation parts, which a clinical trial employee should be aware about. So it is better if you remember these parts and if you correlate like which part is for specifically for things. So part 314 is for new drug application to release into the market. So part 600 and 601 is for your biologicals, and part 812, 813 and 814. So let's go back and check. So part 800. So it's in here. So it's about medical devices. So 812 investigational device exemption, and 814 pre-market approval of medical devices, and also 812 investigational this one, and 813 is reserved. Maybe they are doing some updation related to this particular 813, and then 814 is for your pre-market approval of medical devices. And under title 45, so in case if there are different sort of studies again, so in case if they are participating in public related informations. So let me go back to the home page. So title 45 will describe and explain about the public welfare. So clinical trials are also related to public welfare, and in that it is specifying part 46. So part 46 about the welfare items that we need to look for the part 45. Part 46. So part 46 is standing for protection of human subjects, because informed consent form, everything is also protection of human subjects. So the basic human research subjects, when you are using the details and what sort of regulations you need to follow when it is falling under public welfare. So it's important to remember all these parts of Code of Federal Regulations. And let's see about in-depth knowledge in the Institutional Review Board and Independent Ethics Committee. So Independent Ethics Committee and Institutional Review Board, they both has similar activities, but only the name is different. Both are called ethics committees and both have same rules and regulations, same set of applicable activities and responsibilities. But if an institution is having a review board, so for example, Manipal universities. If you see, there is a Manipal hospital, Manipal colleges, and also Manipal sites. So Manipal hospitals are associated with an institution. So in case if a clinical trial is conducted at Manipal hospitals, they will have an institutional review board than an independent ethics committee. So if you take an example of Apollo hospitals are not connected with any institution. So they are going to, it's an independent hospital. So they are going to have an independent ethics committee. So that is the only difference. It is an institutional review board. It is an independent ethics committee. Both are ethics committees. So we will see the introduction, composition, responsibilities, functions and procedure of IRB and IRC. Okay. So let us see the definition and purpose of institutional review board. So institutional review board is also known as an independent ethics committee in some countries. It is a committee constituted of medical, scientific and non-scientific members as well as community representatives. IRBs are typically established within academic institutions, hospitals or research organizations. So we were telling that ethics committee has to approve, they need to review the protocol, and only if they approve, then we can conduct a clinical trial. So this ethics committee will consist of a group of people who are actually sitting in a conference room and reviewing the documents. So this committee will be provided with protocol, case report forms, and also informed consent documents. All some basic essential documents will be provided to them. And they're going to sit and review this information. And once they review, then they're going to approve or reject the clinical trial. So the main purpose is the primary role of an IRB is to review, like reviewing all the documents, approve, and monitor the research involving human participants. So not only approving, once it is approved, after that, until a clinical trial is happened, until the clinical trial is happening, the ethics committee is going to monitor the progression of this clinical trial at clinical study site. So how are they going to monitor? Every month there will be a progress report submitted to the ethics committee by site staff. That is, your investigator has to submit the progression report saying that the 10 subjects have come and they have experienced some 15 adverse events and another concomitant medication is given and they're doing good and they are being asked to come for the next visit. So this is the status and it has to be submitted in a document to the ethics committee. So ethics committee is going to ask questions like, what are the adverse events? Give me more information. Then again, investigator has to write and send the details of the adverse events. So that's how an ethics committee is going to monitor to ensure that it is conducted ethically and in compliance with the regulatory requirements and with adequate protections of the rights, safety, and well-being of the participants. So ethics committee is going to oversee if the rights, safety, and well-being of the participants is correctly maintained or not. That is the main purpose of the having an ethics committee. And let's see the independent ethics committee. So independent ethics committee is a committee which is similar to an IRB, but typically established outside of an academic or research institution. As I've given an example of Apollo hospitals and Manipal hospitals. So Manipal hospitals have an institution. So they are going to have an institutional review board, wherein independent hospitals will have an outside independent ethics committee. So IECs may also be referred to as in research ethics committees or ethical review boards in some context. So what is the purpose of IEC? It is same like your IRB. So the primary purpose of an IEC is to review, approve, and monitor the research involving human participants to ensure ethical conduct and participant protection is maintained. And also to make sure everything is according to the good clinical practice and according to the regulatory requirements. And the composition. As I have mentioned that there are a group of people who are going to review the study documents. So this group of people is called as the IRB composition. So the number of people or the number of members who are reviewing the study documents. So the IRB and IEC should consist of a reasonable number of members who collectively have the qualifications and experience to review and evaluate the science and medical aspects and ethics of the proposed clinical trial. So anyone cannot come sit and review the protocol. They should be qualified people who has to review the protocol. So we are specifically saying multiple times that without an ethics committee approval, a clinical trial cannot be conducted. So when we are saying ethics committee approval is very important, then the person who's providing approval and the group of people also should be highly qualified. And let us see, this is also called as a quorum. So you can call it as a quorum, group, or reviewers, or also composition. The composition of IRB IEC should include at least 5 members as per US FDA. And it should have at least 5 to 7 members per Indian GCP. So US FDA says 5 members should be present, but Indian GCP says 5 to 7 members should be present. And in which at least 1 member whose primary area of interest is non-scientific area. So meaning like, see, if everyone is from science background, they cannot look the bird eye view of a normal person. So that's why in this group of 5 to 7 people, we are having a layman. So layman is nothing but who is not from the science background. So you can take a lawyer, or you can take an accountant who's working in the bank, State Bank of India or HDFC Bank. We are taking them, and they are also sitting in part of this discussion where to approve or not approve a clinical trial. And they will also be explained about the pros, cons, what is the protocol is all about, because we want to take the opinion of the non-scientific area person as well. So not only from science people, we are also taking it from non-scientific person. And at least 1 member who's independent from the institution or trial site. So there should be 1 member who's not part of the clinical trial and also not part of ethics committee. So he should be an independent person. So a principal from a college or a separate doctor from any other area, we have to have them as a member. And at least 1 woman should be present in the quorum. He can be from either scientific or non-scientific background, but there should be a woman in the group where they are approving or rejecting a clinical trial. So 5 to 7 scientific members, when we say, there can be a pharmacist, there can be a PhD scholar, there can be a doctor, there can be a dean who's dealing with a college. So anyone can attend it. So this is the composition of the ethics committee. And only those this ethics committee members who are independent of the investigator and the sponsor of the trial should vote for the opinion. So an investigator and sponsor cannot vote or cannot approve their clinical trial, right? So an independent body who is part of ethics committee has to vote for the opinion. And a list of our ethics committee members and their qualifications should be maintained. So there will be a document where it will be maintained on about this particular composition. Now let us see the responsibilities of the ethics committee. So an IRB IEC should safeguard the rights, safety, and well-being of all trial subjects. So special attention should be paid to trials that may include vulnerable subjects. So we have seen the definition of vulnerable subjects. So if any vulnerable subjects are used in a clinical trial, special attention should be given to those particular clinical trials from ethics committee. And the ethics committee should review a proposed clinical trial within a reasonable time. So you cannot take how many days you want. When a protocol is submitted, ethics committee should have a reasonable time and document to review and clearly identify the trial. The documents reviewed and the dates for the following. So either you have to give an approval or rejection, or you are giving a favorable opinion. So favorable opinion meaning like, see, I'm okay with your protocol, but I have some questions. So if you answer these questions and if I get a proper justification, then I will approve your clinical trial. So that is called as a favorable opinion. So where it is not an approval, not an rejection, but they are waiting for the answers from the sponsor. And ethics committee can also suggest in case if there is any modifications required prior to its approval. or ethics committee can directly disapprove the protocol. Or ethics committee have an authority to terminate the study happening in across the country. So because if protocol is not scientifically sounding and if there is any mistake in the protocol, ethics committee can take a decision of terminating of this study. Prior approval, they can terminate it. And the continued responsibilities, the ethics committee should consider the qualifications of the investigator for the proposed trial. So as documented, so we are going to collect the curriculum vitae, the current curriculum vitae, and also the medical registration certificate of the investigator. So investigator might say that I am a doctor, but ethics committee wants to review the background of the investigator and also the medical registration certificate and documents related to his education and the current work. So ethics committee would require these documents. An ethics committee should conduct continuing review of each ongoing trial at intervals. So either it can be a monthly report or quarterly report or a half-yearly report. But at least once in a year, an ethics committee should review a report from the site. Ethics committee may request more information than mentioned in the essential documents like a protocol, case report forms. If ethics committee needs more information, they're going to ask the sponsors and they need to provide an elaborative information to the ethics committee. In the judgment of ethics committee, the additional information would add meaningfully to the protection. So why the ethics committee is asking ethics committee? Because they want to protection of rights, safety and well-being of the subjects. So when a non-therapeutic trial is to be carried out with the consent of the subjects, a legally acceptable representative. We discussed about LAR and also impartial witness. So there is a requirement in case if the subject can't give a consent, we would require LAR and impartial witnesses. So ethics committee should determine that the proposed protocol and other documents adequately addresses relevant ethical concerns and meets applicable regulatory requirements for such trials, where they would require an LAR. In case if the site is doing an infant trial or a child-related trial or a vaccination or a drug-related trial, where only kids, it's for kids, it's a medicine given to the kids, then obviously there is a requirement for LAR. So each informed consent form should have signatures of the LARs, legally acceptable representatives as well. Then ethics committee, where the protocol indicates that prior consent of the trial subject or the subject's LARs is not possible, then ethics committee should determine that the proposed protocol or other documents adequately addresses relevant ethical concerns and meets applicable regulatory requirements for such trials. So in case if LAR and if participant consent is not taken, there are some clinical trials where they want to test the trial and where the medication would have been already an existing treatment. Then we call it as an emergency situation where informed consent is not taken and they need to intimate the ethics committee. And ethics committee should determine what is the purpose and why they are collecting like that. So the ethics committee should review both the amount and the method of payment to subjects. We told that in informed consent form that we are going to pay to subjects. So when we say we are paying to the subject, what is the method? Are we doing an online transaction or are we giving a gift cards? Or are we giving cash to the subjects? Because many hospitals, investigator will keep the money instead of giving it to the subjects. So ethics committee will make sure that there is a receipt and there is a signature of the patient as well on the receipt, that the amount and the method of payment to the subject to assure that neither presents problems of coercion or undue influence on the trial subjects. So payments to a subject should be prorated and not wholly contingent. So for example, if a patient receives 1 lakh rupees for a clinical trial, okay, 1 lakh rupees. And if each visit is paid 10,000, okay, so if each visit subject is paid 10,000, then how many visits would require for you? So let me do a math calculation. So if there are 10 visits and per visit subject will be paid 10,000. So the whole amount that we are paying to the subject is 1 lakh rupees. So here, whenever the subject is completing each visit, we are paying him the amount. We are not giving him 1 lakh at once. We are not giving him 1 lakh even at last, or we are not giving him 1 lakh in the start. If the patient has completed 3 visits, then he will receive 30,000. 4 visits completed, 40,000. So this is called prorated. Depending on the visit the patient is completing, he will be receiving the amount. So always the payment to the subject should be prorated, and we are not giving wholly contingent amount to the patient. The ethics committee should ensure that information regarding payment to subjects, including the methods, amounts, and schedule of payment to trial subjects, is set forth in the written informed consent form. So the payment related information should be made available in the informed consent form. And any other written information to be provided to subjects. If you are providing any other information to subjects, it has to be documented in the informed consent form. And the way payment will be prorated should also be specified in the informed consent form. Then let us see the main functions of the IRB and IEC. So the ethics committee should perform its functions according to written operating procedures. So ethics committee will also have a standard operating procedures, and as per their standard operating procedures, ethics committees has to maintain and continue the functionings. They should maintain written records of its activities and minutes of its meeting, and should comply with good clinical practice. So whenever there is a discussion within this group of people or within the composition, always there should be minutes of meeting, meaning like whatever has been discussed in the meeting and what decisions have been made in the meeting, how many people are present and what are their designations, everything has to be recorded. So that is called as MOM, minutes of meeting. And everything has to be recorded, and even ethics committee should follow good clinical practice and applicable regulatory requirements. So an ethics committee should make its decision at announced meetings. So you cannot randomly take a decision whenever an ethics committee wants. So they should inform that there is a meeting happening and ethics committee is having a meeting, and there is a quorum of people who are attending it, and only then they can announce a decision that they are taking. And as stipulated, the ethics committee should have a written standard operating procedures, and they need to follow this operating procedures. And if ethics committee is not following this standard operating procedures, an investigator or a study site can complain that ethics committee is not following the required and applicable regulations. Only members who participate in the ethics committee, IRB and ethics committee, and discussions should vote for their opinion. So whoever is participating in this, so the composition people only has to vote for the opinion, either approval, rejection, or favorable opinion. Approving a clinical trial, rejecting the clinical trial, or giving a favorable opinion for the clinical trial. The investigator may provide information on any aspect of the trial, but should not participate in the deliberations of the ethics committee. So investigator can provide the information to the ethics committee, but he should not be part of ethics committee meeting. Why he should not be part of ethics committee meeting? Because we don't want that bias. Ethics committee is an independent body. Okay, it's a sort of an audit, outer external people who are looking out and who are conducting things on this particular clinical study site. So anyone from your clinical study site cannot be part of the ethics committee. So an IRB IEC may invite non-members with expertise. So because if you remember, we have seen we would require a non-scientific person, we would require a woman who has to participate. So an ethics committee can call out for this non-members, And they can give an opinion on this protocol. They can study and they can also vote for it. And these details have to be added into their composition details. And what are the procedures that ethics committees will follow? So the ethics committee should establish, document in writing. So whatever they are discussing, okay, it cannot be verbal. If there is any question, again, they cannot call the sponsor or they cannot call the investigator and ask the question. Everything has to be in the written format. They need to write the question and they need to send it an email. And then it follows the procedures and then your sponsor will respond to it. So not only questioning, even anything that ethics committee is taking part. When they are saying ethics committee is having a composition. So the composition of the ethics committee has to be highlighted. And there should be a document where it explains the details of the members and their designations. So the ethics committee has to determine its composition. So the document should have the name of the member, whoever is reviewing, and the qualification. What is his background? And whether is he eligible or not? It should be highlighted in the composition document. And this composition document will be reviewed by sponsor also and also by the investigator. If they feel this ethics committee is not having a proper reviewers, now the hospital can choose any other ethics committee instead of one ethics committee where they don't have a proper composition. So even the site staff can choose a different ethics committee, a different body. So the other procedure is scheduling and notifying its members of and conducting the meetings. So ethics committee should conduct monthly and 15 days once or quarterly meetings to assess the progress of the clinical trial. So when we say investigator has to submit a progress report of clinical trial to the ethics committee should also have a meeting. So before this meeting, investigator has to submit it. And during this meeting, there is a conference room where everyone who is part of the composition are going to sit and discuss about the progression of this particular clinical trial. So any document or report submitted to this particular people called ethics committee will be reviewed during the meetings. So conducting initial and continuing review of the clinical trials. So during the initial also they are approving, they are reviewing, and they are continuing this review throughout the clinical trial until the clinical trial is completed. And also ethics committee has to determine the frequency of continuing review as appropriate. So ethics committee can anytime ask investigator site to provide an additional information on the required clinical trial protocol. They can anytime ask about the progression and any questions can be raised in between. And there is an inspection that anytime an ethics committee can come and inspect the clinical study site. So ethics committee providing and according to the applicable regulatory requirements, expedite the review and provide the opinion. So in case if a clinical trial requires an immediate approval, so and if the next meeting is happening in the next month, so then the investigator site and sponsor can request this ethics committee to expedite the meeting and to review the clinical study protocol. So when we are requesting for expediting this review and then providing an approval or favorable opinion, ethics committee should provide that support and they should conduct in the favorably expedite requirements of the study sites. So again, the reason for the emergency review or expedite review should be properly justified. So if there are any minor changes, we call these changes as amendments. So if there are any changes in the protocol, CRF or informed consent form, ethics committee has to inform the clinical study site. And clinical study site is going to inform the sponsors. And now the sponsor will sit and work on the ethics committee suggestions. Or again, sponsor and ethics committee will have a call. And in this call, they can discuss about the suggestions, pros, cons, and what has to be changed and why it has to be changed from the ethics committee perspective. When these sort of discussions are made, essential documents like our protocol, case report forms, and your informed consent forms, there is a chance of changing the information that is captured in this document. So we can change the information during or when an approval is receiving or when a clinical trial is on a conduct phase. When a clinical trial is conducted, till that time also, if there are any suggestions from the ethics committee to change depending on the safety and efficacy aspects of the clinical trial, the changes can be made in the protocol or any other study related document. So ethics committee specifying that no subject should be admitted to a trial before ethics committee issues its approval form. So before an ethics committee approval is received, you cannot start a clinical trial at site. Only when an ethics committee approval is received, after that, a clinical trial can be started. Till that, the site can prepare like how to conduct a clinical trial. They can start having the training procedures, but they cannot conduct a clinical trial. They cannot start informed consent procedures as well. They need to wait for the ethics committee approval. Only when ethics committee approval is received, then a clinical trial can be initiated and a clinical trial can be started. So the other procedures continued. So ethics committee specifying that no deviations from or changes of the protocol should be initiated without prior written ethics committee approval. So deviations are nothing but any misconceptions or misleading in the conduct site. So if protocol says that you need to follow so and so item, if the study site is not following it, then that is called a deviation. So before ethics committee approvals, none of the deviations or protocols can be initiated in the site. So except when necessary to eliminate immediate hazards to the subjects or when the changes involve only logistical or administrative aspects of the trial. So change of monitor, change of telephone number, or change of the point of contact, or change of study nurse or any other site staff, that is okay. But you cannot start with a clinical trial without the study protocol approval. You cannot change anything also. In case if a document is submitted to the ethics committee and now you cannot change a document and use it in your study site. If you are changing, again it has to go to the ethics committee review. So specifying that investigators should promptly report to the ethics committee. So if there are any, once an approval is completed, if there are any deviations or if there are any changes in the protocol or if there is any to eliminate any immediate hazards to the trial subjects, ethics committee has to be notified. Any changes increasing the risk to subjects or affecting significantly the conduct of the trial, ethics committee has to be updated. All adverse drug reactions that are both serious and unexpected. We have seen the categories for serious adverse drug reactions as well. So an adverse event and adverse drug reactions and serious and unexpected. Everything has to be notified to the ethics committee. Any new information that may affect adversely the safety of the subjects or the conduct of the trial should be informed to the ethics committee. So ensuring that the ethics committee promptly notify in writing the investigator and institution concerns. So in case if there is any investigator concern, then ethics committee should promptly notify it. So if there is any trial related decision and openings, we have to notify them. And the reason for decisions or opinions that are taken, you have to inform. And the procedures for appealing the decisions and opinions, it has to be informed. So if there are any, so it should be mutual. Investigator has to inform and also ethics committee has to inform if there are any changes or if there are any specific modulations to the protocol. See, ethics committee is involved a lot. So there is so much of involvement of ethics committee in the clinical trials. So we have seen about the importance of IRB IEC, and we have also seen about the informed consent form, case report form, and also protocol. And we have seen institutional review board, ethics committee, the importance of the definition, responsibilities, composition, functions, and procedures of informed consent form.